From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 62, recorded on November 25, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, great to be with you from LD Lab Studios, or TWiEVO West headquarters,
TWiEVO West.
I guess.
That's a good name.
Yeah.
How are things with you, Nels?
Things are good. So, instead of the weather, we've usually been talking about kind of coronavirus forecasting a little bit. And we might be bracing a little bit for a spike as the Thanksgiving holiday is coming up. Some troubling signs in terms of just the numbers here and some of the indications that people are sort of kind of continuing their holiday plans, and so trying to hunker down and take it easy. How about from there out in TWiEVO East?
It's business as usual, couple days at home, couple days in the lab, mostly podcasting. Waiting for a big increase post-Turkey Day. We'll see what happens. But we have a guest today for your listening pleasure. He's going to talk about a topic that we've had a couple of times here on TWiV. He's a professor at the Rockefeller University, not far from me across town, Daniel Kronauer. Welcome to TWiEVO, Daniel.
Thank you. Hello.
Great to see you, Daniel.
In normal days, you would have been able to come to the studio, or I could go there and record. But these are not normal days, as we know.
Yeah, and in fact, so on TWiEVO episode 18, a couple of trainees in Daniel's lab did come to TWiEVO East. I was hosting another trainee or former technician from Daniel's lab here at TWiEVO West, Sylvia Chang. And then if you remember, you had Buck Tribble and Sean McKenzie in studio for a discussion on Raiders of the Lost Orco, talking about clonal raider ants and genetic manipulations. One of my favorite titles of a TWiEVO episode that you came up with there, Vincent.
It is good. Well, it's all,
that's true.
All based on the
That was like 2 years ago or 3 years ago almost.
Yeah, and we'll have to get an update from you on sort of current advances in clonal raider ant genetic manipulation and studying the behaviors.
Yeah.
But that was if I remember, that was the first manipulation of a gene in ants. Is that correct?
Yeah, that's correct.
Okay. And you must have done more since then, right?
We've done more since, yeah. The hard part was really figuring out the biology.
Yeah.
Right? Like ants are not organisms that had been manipulated in the past genetically in that way. And so we had to figure out all the injection protocols and the raising the eggs, raising the larvae. All of that is really hard because usually that's what the ants do, right? And so those protocols didn't exist. It was really like getting a handle on the biology.
Yeah.
And now that we can do that fairly routinely, of course we can do all kinds of stuff with the ants now. We have a few transgenic lines now where we put stuff in rather than knocking things out. And it's still nowhere as fast as you can do these things in flies or something, but at least we can do it now in principle, which it's nice. It opens up a lot of stuff. And the three people you had in the studio, they're all doing very well still. I think Sylvia is at Cornell now. She moved with one of your colleagues.
That's right, with
Cedric. Yep. Yeah, exactly. Yep. Just working on transposons with him for a PhD. And Sean is working with Oxford Nanopore, actually. He's, I think, test driving the newest technologies there. And Buck is at Harvard now. He has his own lab.
Nice.
Oh, wow. Very cool.
Is he
still working on ants?
On ants, yeah. Very good.
Yeah. Cast development and ants is his focus.
My favorite part of that was the magic marker assay, right? Where you draw on a filter paper and the ants don't cross it, right?
Yeah. That's an essay we sometimes demonstrate at kind of science outreach events at Rockefeller.
That's perfect.
You can do it with the ants. They get repelled by these markers, and you can also do it with termites. And the termites actually follow the markers. They use it as a trail pheromone almost.
And so the orco disruption then made them not able to smell, right? So they crossed it.
Yeah, exactly.
That's just
So
great.
then they don't care about it anymore, and they're blind, so they don't see it. It's all smell.
Yeah, they're blind to begin with, right?
Yeah.
Great. Well, what brings Daniel here, besides ants, which is very cool, he's written a book, Army Ants, Nature's Ultimate Social Hunters. So we'll chat a little bit about that. But first, let's probe a little bit into your background, Daniel. Tell us how it all began.
All the way back. I'm from Heidelberg, which is like this quaint little college town in Germany, kind of a medieval town, but it's kind of a famous university and a lot of science going on there. There's a lot of Max Planck Institutes. There's a German Cancer Research Center. So that's where I grew up. And that's also where I started studying biology. And I think I fell in love with biology as a little kid. Like I always had a lot of exotic pets in my room, and like anything from chameleons and scorpions and all kinds of stuff. And of course also like ant colonies, a lot of different kinds of insects. And
so Daniel, did you actually have those like little ant farms where it's the clear kind of plexiglass and you can see them building their little tunnels?
I had them later. Like I actually started out completely ignorant about anything. I took like my little sister's old kind of diaper cardboard boxes and caught like bumblebees in the garden, put them in there. And of course that didn't work at all. That was when I was like 6 or something. I had nobody to give me really advice on how to maintain these things. But then later I became more professional, and ultimately I ended up with like real ant farms and stuff. But so then I started to study biology in Heidelberg. And then after my pre-diploma in Germany, you do 2 years of kind of undergrad studies, and then you kind of specialize more. And after my pre-diploma in Heidelberg, I then moved to the University of Würzburg, initially with the intention to study tropical ecology. I was really into the tropics. And I think like a lot of organismic biologists are just fascinated by all the biodiversity that's out there. And the University of Würzburg has, I think, the only tropical ecology department in Germany.
I was going to say, you don't necessarily put Würzburg together with tropical landscapes, and so, but the scientists are there, I guess.
Yes, yeah, and they run a big research station in Comoy National Park in Ivory Coast. That's like their main study site, but they also do work in Southeast Asia and tropical America, and so that's where I moved, and then. From there, I spent a year at Duke University in North Carolina as a non-degree graduate student, like a visiting graduate student. And the connection there was that also at Duke University, there's a lot of tropical ecology, tropical biology going on. The Organization for Tropical Studies has their headquarters there, OTS. And so I took classes there with people like John Terborg. He's a famous tropical ecologist. But then while I was at Duke, got more and more interested in population genetics and molecular phylogenetics, and more and more into kind of evolution and using molecular markers to study evolution. And then when I came back to Würzburg after my exchange year, I really wanted to go into population genetics. And then the only person who was really doing that there was Jürgen Gado, who was kind of a junior faculty in the department of Bert Höldobler, which was kind of the neighboring department to the tropical ecology department. They were working on social insects. It was the Department of Behavioral Ecology and Sociobiology. And so then I kind of switched gears a little bit and really got into ants, and started working on ant population genetics with Jürgen and Bert. And then, of course, also kind of realized that, even if you really want to do work in the tropics, ants are actually great because there's tons of ants in tropics, and you can actually just pick tropical ants to work on if that's what you want to do. And that's what I did for the longest time. So then initially I did my diploma thesis actually on the evolution of slave-making behavior in honeypot ants. Those are ants that live in the deserts of the southern US and northern Mexico. And so did field work there in New Mexico and Arizona, Baja California. And then did population genetics and kind of using genetic markers to assign ants to the colonies that they were born in, and then following ants as they were kind of stolen across colonies and enslaved by other colonies.
Did you connect at all with Jack Longino, my colleague here at Utah, with any of this work? Or was that?
Not back then, but I know Jack very well. He obviously is kind of a very famous taxonomist. He did a lot of work in Costa Rica, where I then later did some field work, and actually where a lot of the stories in my book are centered on, because that's where we did a lot of field work on army ants later. And Jack spearheaded kind of an inventory of the ant fauna in Costa Rica, which is probably the most complete inventory of any tropical ant fauna. So he was in charge of that. And yeah, he just described a new ant species from his garden in Utah, actually. So that's kind of a neat discovery. So then while I was doing my master's thesis in Würzburg, I met Koos Boomsma, who was there on sabbatical. He's a professor at the University of Copenhagen, and his kind of main focus is leaf cutting ants, fungus growing ants, and those are neotropical. So those are ants that occur in the New World tropics. And so I started to talk to him a little bit and got really interested in his work, and initially thought maybe I could join his group as a PhD student to study leaf cutting ants, but then he suggested that maybe I should focus on army ants instead. So he knew that I wanted to do a lot of field work in the tropics, and that is a group that back then was really not studied at all using population genetic markers. So I kind of really realized that the opportunities would be enormous. Army ants are like a big group of ants. They're quite substantial. They're in a separate subfamily, the dorylines. And they're ecologically extremely important, as I guess we'll talk about a little bit more later. But so that seemed extremely attractive, and those ants have like an amazing biology, amazing life history, and so then for my PhD I moved to the University of Copenhagen to work with Koos on army ants, and that's kind of where the whole army ant story started. And then did a lot of field work in mostly Venezuela back then, and also in Kenya on African army ants. And then after my PhD, I moved to Harvard University in Cambridge as a junior fellow at the Harvard Society of Fellows. And so then I was there for 3 years. And that's kind of where I started to develop the clonal radar ant that we talked about in the beginning briefly as a model system. The idea was to do functional genetics and behavioral genetics in ants.
And tell us, could I ask Daniel a little bit about that junior fellowship? So this is sort of like a postdoctoral step, but it's a little different, sort of a super postdoc in some ways. How would you describe it?
Yeah, it's kind of an independent fellowship. But as an experimentalist, you kind of have to associate with some lab. So I ended up working in Naomi Pierce's lab. She's a professor at the Museum of Comparative Zoology at Harvard. The junior fellowship, it's an interesting fellowship to have because it's very broad. So they elect 10 junior fellows every year, but across a broad range of disciplines. So some of them are mathematicians, physicists, but also from the humanities, they're philosophers and people from the English department and political sciences, and of course biologists. And so it's a very, you kind of get to know people from very diverse fields, and it's structured in a way that you meet once a week for dinners, all the junior fellows, so 30 junior fellows across the 3 years, and the senior fellows as well. Usually there are about 10 senior fellows. And then you meet twice for lunches a week, so you get to talk a lot. And
do you think this
Tuesdays you're usually hungover, and you lose a lot of time as well. But it's interesting. It's a nice way to broaden your horizons.
Yeah, no, it sounds like great cross training. So do you think that this influenced your idea to write a book? Actually, hanging out with folks in English and the humanities, things like that, did that have some impact?
It certainly influenced the way I think about problems, or especially how I choose problems. I think the book itself, it's interesting, right? Because in the humanities, I think it's very common to write books. Like you write one book as your thesis, and then one book as a postdoc, and that's kind of your two works, right? Versus in biology, people focus much more on papers, scientific papers, like scientific articles, like research articles, right? Like usually people start writing books when they're older. That's kind of the trend, at least. I always really liked the idea of writing a book. And in my field, at least in myrmecology, it's actually not that uncommon. Like there's this tome that Bert Hölderlin and Ed Wilson wrote, The Ants, from 1990 with Harvard University Press, which is this massive book. And I think it really inspired like a whole generation of myrmecologists. And maybe that's part of the reason why, at least in my field, I think books are still kind of very important, actually.
I'm wondering why you, so you went to Harvard, but you didn't continue to work on army ants. You switched to raiders. Why is that?
So actually, the clonal raider ant is actually a close relative of army ants. And the reason I wanted to switch was mostly that I think towards the end of my PhD, I don't want to say frustrated, but I realized that army ants are just very, very hard to work with, right? They have huge colonies, like hundreds of thousands of individuals, and you can't keep them in the lab. So you can't really do controlled experiments. It's all fieldwork based, and so you're a little bit like a researcher studying a group of chimpanzees or something, right? That you follow through the rainforest. Like every night they move, you have to be there, you have to follow them, and even like just studying more than maybe 2 colonies simultaneously is almost impossible, right? Because you have to be in the forest every night to follow those colonies around so you don't lose them. And so for population genetics, that's fine, right? Because you just collect your samples in the field, you bring them back into the lab, and you analyze them. That's actually kind of nice. But if you want to do behavioral experiments and eventually functional stuff, that's just really hard. And it seemed also, in terms of running a lab working on army ants, that didn't seem very realistic. And the other thing is that I think I became a little bit frustrated also with the fact that generally in the field, people tend to pick one species that they work on because of a certain problem they're interested in. But usually you can't really maintain most ants in the lab for very long. So people go out into the field and they collect new colonies every season. And so you never get really good control over the genetic background. You can never repeat the same experiment with the same genetic background, or even a different experiment with the same genetic background that you can really compare it to previous experiments. You can't breed ants in the lab usually. You can't propagate them. And as a consequence of that, you also can't really do functional genetics because you can't maintain transgenic or knockout lines easily. And so there's all these like biological kind of limitations as to what you can do with ants. And actually the same kind of applies to honeybees as well and other social insects. And so I really kind of took a step back and started thinking about whether there would be a species where we might be able to kind of crack those barriers and maybe you move beyond the kind of stuff that people have been doing so far. And from having read all this literature around army ants, I knew about the clonal raider ant, which back then there were only, I think, maybe 2 or 3 papers published about the species. And in kind of a more cryptic social insect journal. But those 2 papers basically suggested that first the species was clonal, and so you could propagate lines without needing to cross anything, and potentially you could put in transgenes or knock out a gene and then just propagate that from a single individual. They don't have queens, so like all the individuals in a colony can reproduce, which makes things much easier. And those and a few other important biological aspects of the species that just seem very useful for a model system. And so then I switched gears a little bit. But we can still use the species actually to learn a lot about army ant behavior, which kind of makes it especially nice from my perspective.
No, it seems like it was a great pivot. And as you can hear on episode 18 of TWiEVO, I'd point all our listeners there. And then with that update, sort of where are they now? It's just continuing to grow in great ways. So then that was 3 years, but then you transitioned to take us through that next step, that transition to opening your own independent laboratory.
So my idea was, or at least what I proposed to the Harvard Society of Fellows, is that I would start developing the system as a model system for social insect functional genetic stuff. And of course, as like a single postdoc without a lot of resources, that turned out to be pretty much impossible, right? So then actually during the 3 years, I mostly focused on establishing rearing protocols for the species in the lab, and just growing up colonies, and then doing a few first population genetic studies simply to show that the species is in fact clonal, and then looking at kind of the species is invasive actually, so it occurs on like tropical and subtropical islands around the world, and so trying to figure out where the species actually comes from, like how much genetic diversity is out there in the invasive range, how is it distributed, those kind of studies, and then also doing a few first behavioral studies to kind of take advantage of this genetic control that the species has. So that's kind of the stuff I did then doing my postdoc. And then when I started applying for faculty positions, really I had at least some data in mind that was kind of proof of principle that the stuff I wanted to do was, at least in theory, possible if I had the appropriate resources. And luckily Rockefeller University believed that I would be able to maybe do it. And so they gave me a job, and since then I'm here, moved to New York, across town from Vincent. As we already said. And,
So how many years now are you at Rockefeller?
So I moved to Rockefeller late 2011.
Okay.
So I'm pushing 9 years.
And there you were able to establish the clonal raiders as a system that you could work on, right?
Yeah. So now initially when I started my lab, it was still a little bit mixed. So I would say maybe half of the people were working on clonal raider ants. And then the other half, I still had some people working on army ants and some other ant projects, partly because I wasn't sure whether this thing with the clonal raider ant would really pan out the way I hope it would. But now everything is, at the moment at least, everything is clonal raider ants, because now we can really do a lot of stuff that really opens up a lot of possibilities. So we kind of came from like a number of approaches, because it's a little bit of a study system centered lab, right? It's less of a question centered lab, but basically we're coming from the species, but from all kinds of different angles, and study anything from like communication to development, and now kind of start to move into circuit neuroscience to understand kind of circuit basis of social behavior. And so it's a lot of kind of different questions. And so it's a nice lab. It's very diverse, with people with, a background, anything from stem cell neuroscience to epigenetics and circuit neuroscience. And it's very diverse, which is kind of a nice group to work with and get ideas, and people complement each other, and there's a lot of collaboration going on.
I'm curious about how we talked briefly about the book, but tell us a bit about how it actually came to be.
The book. So initially, actually, what happened is that towards the end of my PhD, suggested, my PhD supervisor, he suggested that I stay for another year and write a book about army ants. And I really loved that idea, and I agreed to stay. But then kind of it dawned on me that I wasn't really yet at a stage where I would be able to write the kind of book I would want to write. And so then I stayed, but it was more to finish up experiments, start a few new ones, and get out a couple more papers for my PhD. But since then, I've had that idea in the back of my mind that one day I would want to write a book about army ants. And then I was at the meeting at Harvard, actually, for the birthday of Naomi Pierce, who was the person I worked with doing my postdoc there. And there was somebody there from Harvard University Press who then contacted me afterwards. She had seen my talk there and asked whether I would be interested in maybe writing a book with them. And then, of course, this idea of eventually writing an army ant book came to mind. And so I discussed that possibility with her, and then eventually decided to just go for it. And that was at the time when, my lab had developed to a point where I felt kind of confident that things were running now. And I wanted to have a project that I could work on by myself a little bit. And it was hard to do experiments in the lab, because if you're the PI and you go to the lab, there's no way you get anything done. And so with this, I could lock myself up in my office or do it at home at night. And so it was this kind of side project that then, of course, like dragged out over a few years. And but it was also nice because it complemented a lot of the projects that were going on in the lab, and it forced me to read a lot and kind of synthesize ideas that are out there, and that informed a lot of the stuff that we're doing in the lab. And there was also one big project in the lab going on with a postdoc, Christoph van Beer, and we went to Costa Rica, to La Selva, this field site that I already mentioned, basically every year for like a few years to study army ant myrmecophiles. So at La Selva has this really nice community of army ants. It's really very diverse, and army ant colonies host this microcosm of crazy myrmecophiles, like guest species, arthropods that have adapted to live inside the ant colonies and kind of crack the communication code of the ants and are able to like infiltrate the ant colonies. And so that was a project that went on for a few years. And so I was in the field every year and was able to take a lot of pictures because we were following these ant colonies for weeks on end. And so I had a lot of material for a book at that point.
Yeah, and the photography is great. And I guess these are all your photos that you've included. Yeah. So both the writing and sort of the artwork, really impressive. Do you still make it to La Selva from time to time, the field station?
I haven't been in a while. Well, the last time I went was in 2017. That was the last time. And now this year, of course, there was no way of going anywhere, right? But I hope to go back eventually. Like now, especially, I think we're getting more and more. The more we learn about the clonal raider ant and like how the biology of that species is kind of plays out on a molecular level more. We're getting more and more ideas of things that you could actually now go back into the field and test eventually. Once you have like ideas for like targeted projects where you're interested in like certain candidate genes, or at least you know what you're looking for. That seems to become more feasible. So I think over the next years, maybe we'll start developing a few more field-heavy projects again.
Nice, yeah. So we should dive right into the book here. So the book is titled "Army Ants: Nature's Ultimate Social Hunters." I would say the timing is good here, Vincent. Sort of holiday season. Mm-hmm. Put a shout out here for holiday gift ideas early and often. I think the book was published in early October. And I'd say it's time to add army ants either to your dreams or to your nightmares. These are really interesting creatures, practicing complex social behaviors, as Daniel has been hinting at, with really massive consequences on the ecosystems that they occupy. And so maybe just, in my kind of extensive, pseudo-extensive research for our podcast, I was on Twitter looking at Daniel's profile, and you call these critters, the army ants, some of the wickedest insects ever to roam the planet. Maybe introduce us a little bit to these wicked insects.
So, I call them wicked because they have this, they're kind of infamous for kind of attacking and devouring everything that's in their path. And that's of course not entirely true, but like most army ants are actually specialized ant predators. So they really specialize on attacking other ant colonies. But some species have evolved broader diets, and the colonies can be massive. So the species we study in Costa Rica, they can have hundreds of thousands of individuals. Some species of African army ants, estimates go up to about 20 million individual ants in such a colony. So they're really massive. And those are... The ants hunt in big swarms. At least some of these large colony species hunt in massive swarms. So you can walk through the forest in Costa Rica, and suddenly there's this carpet of ants that's like 20 meters wide and 5 meters deep that kind of sweeps through the forest. And these ants the army ants that hunt in these swarm raids, they're also called driver ants, because in front of the swarm raids they drive away arthropods like spiders and cockroaches and crickets that you can see like scurry through the leaf litter and like hop away from the ants and in panic kind of. So it's just like interesting like sound landscape even when you encounter swarm raid. But there's like all this the rustling sound of the fleeing insects. There's the ants that kind of like go up in the vegetation and let themselves fall down back onto the forest floor. It's like this, sounds like a little drizzle of rain or something. There's all these, there are birds that follow the swarms that feed on the on the insects that escape from the ants. You can hear those birds. They kind of vocalize, and they're almost oblivious to observers being there. So you can get really close to these birds, which you usually can't because they're so focused on the ant swarm. There's all these parasitic flies that hover over the ant swarm and that buzz around at the rate front. Those flies also dart down at the fleeing insects, deposit eggs and larvae. So it's really quite a spectacle. It's like, I would say, probably one of the most amazing things you can see as an organismic biologist when you go out there in nature.
Yeah, really interesting.
But these ants, they've made it into historical accounts and even kind of fictional accounts, and I think that's where a lot of their infamy comes from. There's like reports of, for example, African army ants stripping a chained crocodile to the bones. So as long as you can escape as a human or as a mammal, you don't really have an issue. But if you can't, it can become a problem. And so there's, like in South American literature, there's accounts of army ants killing people and newborn babies. These things are exaggerated, but
when you were
that's kind of their reputation.
When you were on field trips, did you ever get attacked by these ants?
Oh yeah, all the time. You get used to it. So I wear high rubber boots, and that kind of can help fend them off a little bit. And the crux about when you work with army ants, you can't move. That's they get very excited if there's movement, and then they attack. As long as you stand still, they kind of ignore you. But that's kind of an art you have to practice, because your first instinct is, of course, to run around when you see that the ants are coming up your boots, right? And that's the last thing you want to do.
Some good side effects too. Aren't these guys really good housekeepers? If they get into your home, you don't need to panic necessarily.
You have to maybe leave your house for a couple of hours. But as you say, the nice thing is then if a swarm like this sweeps through your house, they drive out all your cockroaches and what other kind of vermin you might have, and so your house gets cleaned. Essentially, you have like a free cleaning service if the ants come through.
Yeah, so we'll go pretty deeply into the kind of biology and behavior here, but maybe to begin, we could just talk a little bit from sort of an evolutionary standpoint. So, when you think about the diversity of ants, when did ants actually emerge in the first place, or what do we know about the origins of ants?
So ants evolved about roughly 100 million years ago in the Cretaceous, so kind of the height of the dinosaurs, kind of around that timing. And you see, you actually find fossils of kind of specimens that belong to modern groups of ants as far back as like about 100 million years in things like Burmese amber, which are kind of among the first amber deposits. There's also some ants that you don't see anymore, what are called stem ants. Those are kind of among the most bizarre ants. Those are all extinct. They are also in Burmese amber. But you already see these kind of modern ants there. So that's around the time when ants arose, probably. It's pretty consistent, actually, with molecular phylogeny dating. And then army ants are younger. Army ants are, maybe like on the order of, like the subfamily Doralini, which contains the army ants, is maybe on the order of 80 million years old or something. And those were kind of the first. The Doralines seem to really have invented specialized mimicophagy, so specialized ant predation. So as the ants kind of then rose to more and more ecological dominance and became more and more diverse and abundant, and at some point that life history seems to really have been very ecologically successful. Mm-hmm. And then the Doralines radiated into a lot of species that then specialize on different types of ants that they hunt.
So, and you mentioned, one of the major kind of behavioral aspects, in addition to the predation, somewhat specific to the army ants, that it's evolved on a few occasions, but is also this sort of social behavior, or in the field, I think this idea of eusociality. Maybe tell us a little bit about the origins of that. Help define that for us a little bit. It's a kind of a big question.
How eusociality evolved? Mm-hmm. Yeah. So eusociality is kind of defined by having reproductive division of labor. So eusociality really means like kind of an advanced state of true sociality. And this reproductive division of labor, you have some individuals in the society, in the colony, reproduce. So in ants, that's the queen. She lays all the eggs. And then you have non-reproductive society members. And in ants, those are the workers. And the workers then specialize on brood care and foraging, nest maintenance, nest defense, and so on and so on. Then another defining feature is cooperative brood care. So again, here the workers are the ones who care for the brood, even though it's not their own offspring, it's their sisters and sometimes brothers when the colony produces males. And then you need overlapping generations. That's kind of the third defining feature of eusociality. And so in an ant colony, you have the mother queen and then different worker generations that kind of overlap as adults and live in the same nest. And eusociality has evolved on a number of occasions across the animal kingdom, mostly in insects. But there are things like naked mole rats, for example, that people consider eusocial, which also live in colonies in the deserts of East Africa. And in insects, eusociality has evolved several times within the Hymenoptera. So that includes the ants and bees and wasps. And but also in things like termites, for example, which are very distantly related from ants, right? Like termites are hemimetabolous insects, so they have these like nymphal stages. They don't have a full metamorphosis, versus all the Hymenoptera are holometabolous, so they go from like egg to different larval instars to a pupal stage, and then the adult stage. And these differences in development actually correlate with kind of different types of eusociality in insects, I would almost call them. So in the Hymenoptera, eusociality has kind of evolved along what I consider a brood care route. So the first helpers at the nest, the first workers, were in charge of caring for the larvae. And so as a consequence, also, all the workers in a hymenopteran society, in ants and bees and wasps, they're all female, because it's only the females, ancestrally at least, that displayed brood care behavior, parental behavior. In termites, it's very different. Like termites, because they have the juvenile stages are very independent. They don't have larvae that have to be fed. The juveniles, the nymphs, they look like miniature cockroaches that just can feed on themselves. And the cockroaches live inside their food. They live in rotting wood. So here, the first helpers actually, termites, they are derived from cockroaches. And here, the first helpers that stayed at the nest were more in charge of defending the nest against intruders and competitors. And so here, the first helpers function as defensive soldiers, essentially. And defensive behavior is displayed by both sexes. And in accordance with that, like in termites, the workers can be male or female. So it's slightly different structures of these societies. Some ant species also have soldiers, but those are usually secondary derivatives. So soldiers in ants evolve much later on the phylogeny and then repeatedly, versus in termites, those are kind of at the evolution of eusociality. You get soldier castes. And that's similar in things like eusocial aphids, for example. Those have also soldier workers. Eusocial thrips is the same thing. They have soldier workers. And those are things that kind of nest in galls on plants. And that's also, again, kind of their food source. And it's less about brood care, at least initially, and more about nest defense.
You mentioned that most of the offspring are female, but some are male. So how does that get determined?
In ants?
Yeah.
Yeah. So of course they still have to produce males to procreate, but the males don't play a role in the social life of the colony. They usually get produced in many species seasonally, and then they disperse on the wing. They mate with young queens, and then the males die after mating, and the queens start to found a new colony, start to lay eggs, and then a new colony starts from scratch. The males are produced in Hymenoptera. Sex determination is haplodiploid. So females always diploid, males always haploid, and so the males are essentially produced from unfertilized eggs that the queens lay. And so the queens have kind of control over whether they want to produce males or females by either inseminating the eggs or not.
So, and what determines if a queen goes off and makes a new colony? I mean, if it leaves, then the other colony is gone, right? So how does all that work?
So the queens don't leave their colony, but like in your standard ant, if there's such a thing as a standard ant, I mean, there's more than 14,000 different ants, right? And every one is kind of different and fascinating for its all merits. But like a standard ant, like a black garden ant or something, you have the queen, the mother queen of the colony, and then at some point, say in the spring, she produces a cohort of winged males and winged young queens, and then those leave the colony. They mate. Then the males die. The young queens they go to the ground. They shed their wings, and then they dig a little hole in the soil, and they start to lay eggs. And that queen, of course, then never leaves that colony again. Once the first workers have it closed, the workers do all the maintenance, all the foraging, and so on. The queen just stays inside the nest until she dies, essentially.
How long does the
so it's
queen
only
live?
these young queens that actually leave colonies and disperse.
How long does a queen live?
It depends a little bit on the species, but in some species, the queens can live several decades, up to something like 25, 30 years, which is pretty amazing, right? Because the workers live maybe a few weeks, maybe a few months in some species, right? So there's these tremendous differences in lifespan between the female casts of ants, even though, there's no fixed genetic differences in most cases. It's all epigenetic, all these differences in morphology and behavior, and in longevity, all kind of epigenetic at some level.
And
and also great photos in the book of all of this different morphology. In addition, the queens, the males that you might mistake as not being ants at all with their wings, and
the males look more like wasps and army ants, especially.
And when a queen dies, the colony is finished.
Usually, yes. Usually, the colony is done.
What happens to all the ants? They just wander off and die, or join another colony? No.
So it depends a little bit on the species. In honeybees, for example, honeybees can raise replacement queens. So if the queen dies, they can manage to raise a new queen that then can be inseminated and take over. Mm-hmm. Most ant species can't do that. Some ant species are what's called polygynous. So they have more than one queen, in which case, and they can adopt new queens into the colony. But most ant species don't do that. So, for example, in army ants, if the queen dies, the colony is pretty much done. What army ants do is that queenless colonies, they can fuse with neighboring colonies, so they can join neighboring colonies. So it's kind of this last and final act that they have. But whether they get any real fitness benefits out of that is a little bit disputed.
It's wild. It's just wild. In fact, one of the wildest photos in the book is how the ants make a bridge for other ants to walk on, right?
Yeah, that's like a very army ant specific thing that they do, bridge formation. That's one of the first things that was actually described in what I think is probably the first real account of army ants from the Neotropics by a German naturalist, Maria Sibylla Merian, who wrote a book in 1705 about the insects of Suriname. And so she talks about this, and there's then actually later accounts of people who say this can't be true. There's no way ants do this. So some people at least didn't believe her account of this bridge formation because it sounds so bizarre, but the ants actually do that.
Yeah, no, it's really interesting to see just as the book kind of unfolds that sort of these two major pillars of the evolution, which seems to be reproduction, of course. This isn't sort of a surprise, but then also sort of the predator-prey or the defensive kind of interactions as well, and how those sort of two things intermingle over the life histories of these different species is really interesting. So, and you mentioned a few other species, but it's really cool to just think about the diversity of behavior. So you mentioned, we've already talked a little bit about the farmers, like the leaf cutters that cultivate this fungus. You also mentioned, I think briefly, the slave makers, these ants that will go to colonies, recruit other ants to do the work for them that you were doing for your diploma work. Are there other kind of crazy, notable strategies, social strategies among the ants?
Just a few words about the slave makers. Those are actually, it really took me a while to realize that. So slave making ants, they also raid the nests of other ants. So very much like army ants actually, but instead of then raiding distantly related species and then feeding on the ant brood that they capture, they raid closely related species and then bring the brood back and raise them into workers that they enslave, and then the workers of the slave species work for the slave maker species. And actually, it turns out, so I spent most of the pandemic lockdown in upstate New York in Woodstock, where we have a little house in the forest, and we have one really nice slave making ant colony in our garden of like these North American slave makers. And so I was able to, because I was there all the time anyway. Like over the summer, that's when they do the slave raids, like in the afternoon at around like 3 p.m. is when they usually set on. I was able to watch some of them. And it turns out that they actually, not that unfrequently, also raid the nests of very distantly related ant species and then bring back the brood. And that they can't enslave those, right? They're too far, they're too distantly related. They can't communicate with them. So they must do that as prey, right? They must feed on them. And so it's really actually very reminiscent of army ant behavior in these completely distantly related species, which I found quite fascinating. I mean, I could spend the rest of the evening geeking off about cool ant behaviors, but there's a few other ones that I bring up in the book that I show pictures of, just to give people, in the second chapter where I talk a little bit about ant diversity and the evolution of army ants in particular, to give people like some feeling for what that diversity looks like. So another great life history that's fairly common in ants are these very tight ant-plant mutualisms. And in the American tropics, you can, for example, find these interactions between Cecropia trees and Azteca ants. So these are ants that live inside the stem, the hollow stem of the Cecropia trees, and then they defend the plants against herbivores and also competing plants. And the plant then, in return, they provides housing for the ants, but they also produce food bodies that the ants harvest and feed on. So it's kind of complete board and lodging for the ants in exchange for defensive functions. And that's a theme that has evolved repeatedly many times across ants with different types of ant subfamilies and with different types of plants, similar interactions.
Yeah, and then for the raiding behaviors, maybe I guess, and stepping back a bit, so you mentioned the driver ants of Africa and in the old world. This is all convergent evolution, or what was the common toolkit that was there in place for them to sort of advance these behaviors? How do you think about those kind of questions?
So there's the Doralini subfamily, which has most of the army ants, right? That's where these two clades are, the New World army ants and the Old World army ants. And those have both evolved from ancestors that look pretty much like our clonal raider ants. And so what happened in both cases, and probably independently, there's a little bit of controversy, but I think the phylogeny is pretty well resolved at this point. So probably independently, colony sizes increased. And because of the dynamics that I described earlier, right, like how colony size affects the architecture of these raids, you have this evolution of army ant raiding that looks extremely similar in the two groups. The one issue is that outside of the actual army ant, species haven't been studied very well. There's very few species in the subfamily that are not classified as army ants that have been studied in a lot of detail. Really, the only one is the clonal raider ant. On other species, the observations are pretty much anecdotal, I would say. And so there's a lot of gaps in our knowledge that should be filled in. What's really quite interesting is that there's another subfamily of ants, the Ponerini. And in that subfamily, there's another origin of army ant behavior. And what's really cool is that it has evolved from very similar ancestral behavior, again, from species that send out scouts that then recruit group raids. And then there's just a few species in one genus, Leptogenus, that has large colonies. And those then again from these kind of mass raids of army ants that are strikingly similar to mass raids in the Doralini, to the main army ant clades.
Yeah, that's really cool.
So it seems like, so across the ant phylogeny on several occasions, you had these like pretty almost identical evolutionary dynamics pan out.
So it seems like a major kind of thing to potentially manipulate, and probably impossible. But could you go into the field and try to take like one of these army ant colonies and make it bigger or make it smaller artificially?
Smaller, yeah. So it's a great experiment. I think we should try it at some point. I think it should work. I don't know, but it's hard to tell how much else has been built on top of that over evolutionary time. But the fundamental behavioral rules that they seem to follow are strikingly similar. So I think in principle, there's a good chance that it could work. This transition from the column raiders like Eceton homedum to the swarm raiders like Eceton bucellii that we discussed just a little bit ago, that you can actually sometimes see in the field, because sometimes acetonhamidum colonies just conduct very large raids, or some colonies are just much larger than the average colony, and those sometimes they seem to approach swarm raids. And in very rare cases, they start to attract these kind of swarm-following birds, even that rely on insects being flushed out of the leaf litter and stuff. And at the same time, you can sometimes see very weak aceton bucellii raids. And those then start to look more like column raids. And so clearly there is this kind of graduation that you can actually see in the field. That's kind of a natural experiment, but you could of course do it under more controlled conditions and maybe like cut the colony size in half or double it.
Mm-hmm. So Daniel, you used the word architecture when you described these swarms. And in fact, in the book, I think you call the army ants masters of modular architecture. So what's going on there as we sort of get a little bit closer into the swarm and the kind of mechanics of it or the architecture of it? How would you describe that?
So what's another really fascinating aspect of army ant biology is that they can construct these living structures. Like they're really like architects, but they build them out of their own bodies. And so I would say maybe the most striking one is the army ant nest, the bivouac. So army ants, because they're so efficient hunters, they don't build kind of normal nests like other ants in the soil with like tunnels and nest chambers, but they are nomadic. They have to relocate the colony fairly regularly to move on to new hunting grounds, and so they don't have permanent nests, but instead they build what's called a bivouac, which is this kind of sphere where the ants cling together, a little bit like a honeybee swarm. And but it's a sophisticated structure. It's like formed out of ant scaffolding. The ants interlock their bodies via tarsal claws, so like little claws on the feet. And then there's tunnels inside the bivouac. There's nest chambers in which the brood is housed. They have kind of ventilation walls that they can open and close depending on the ambient temperature and so on. So it's these very dynamic structures. Similarly, they form bridges, for example, when they traverse the leaf litter and there's kind of gaps in the leaf litter. You can often see them form kind of suspension bridges again out of the bodies of the ants themselves, and then the rest of the raid or the immigration columns just walks over the bridge. Like the smallest version of a bridge is what's called a pothole plug. So it's like a single ant that kind of stumbles into, say, a hole in a leaf and it just stays in place. And as long as there's traffic flowing overhead, so there's other ants stepping on the back of the ant, it stays in place and just plugs the pothole.
How often do they move, and what's the trigger for that?
That's it's a good question. So there's very broadly speaking, there's two different types of army ants. There's the ones that are what's called phasic, and then the ones that are non-phasic. And so the phasic army ants, like aceton bucellii, like acetonhamidum, and actually also like our clonal raider ants, they alternate between what's called the nomadic phase and the stationary phase. And in the stationary phase, the queen activates her ovaries, she lays eggs, and there's no larvae inside the colony. And then the larvae hatch from the eggs at the end of the stationary phase. And then it's the larvae that kind of induce the nomadic behavior and increase the activity of the workers and ultimately also suppress egg-laying activity in the colony probably. And so during the nomadic phase, when the colony has larvae, that's when the colony moves. And so basically during the day, they conduct these large raids, and then towards the evening, like at around dusk, the traffic, during the day, there's like bidirectional traffic on the column, right? And the ants retrieve prey. And then at around dusk, this traffic reverses and becomes outbound. And eventually ants start carrying the larvae in the direction of the day's raid along the column that is formed. And then they emigrate. And those emigrations can be over 100 meters long. And then they assemble a new bivouac somewhere else. And from that, they hunt then on the next day. And so during the nomadic phase, the colonies emigrate almost every day. Like sometimes they skip an emigration, but it's fairly predictable, actually. And then in the non-phasic species, it's a little bit more complicated. People don't really know what triggers the emigrations, but they also relocate the nest quite frequently. But in some cases, it can be after just a couple of days of the previous emigration, and sometimes they can stay at one nest site for a month or even longer. So it's much less predictable, and may have to do with, the nest getting disturbed by a predator or getting too dirty, or the ants not finding any food anymore in the vicinity. And then at some point they're motivated to move on.
So when you're talking about phasic behaviors, how many different phasic behaviors are there actually? And how important are these for colonies to operate?
What do you mean by how many different behaviors?
Well, or the definition, I guess, of phasic or versus non-phasic behaviors that you measure.
So a phasic species is just a species that alternates in a predictable way between these nomadic phases and stationary phases or reproductive phases. And it has, these phasic cycles have essentially two components. One is the behavior of the colony, right? So emigrating or not. So during the nomadic phase, they emigrate regularly. They have large swarm rates in Estombucelli. And during the stationary phase, they don't emigrate. The bivouacs are usually somewhere sheltered. They're not in the open like in the nomadic phase. And the queen reproduces. The queen actually she becomes like what's called physogastric. She massively expands the abdomen, becomes pregnant, and she actually can't walk while she's physogastric. So even if the ants wanted to, they wouldn't be able to really relocate the nest during that phase. And it's a really fascinating phenomenon because there's a lot of biological rhythms out there, right? And most of them can be classified as either being endogenous, things like the circadian clock or menstrual cycles or so on, and others are exogenous, things like lunar cycles, right? Many marine invertebrates, for example. But these army ant colony cycles, they are kind of exogenous at the level of the individual ant because they are regulated by signals that arguably come from the larvae. But then at the level of the colony, the colony superorganism, if you wish, they are endogenous, right? Because they're not like colonies in the population, they're not synchronized. So it's not like they do this according to like the lunar cycle or something like that. Mm-hmm. So it's kind of it's a very interesting phenomenon. And for us, it's also interesting because we can use it to learn about kind of the molecular underpinnings of behavioral differences, right? Like what regulates foraging behavior in the ants and what regulates reproductive activity, which are kind of very interesting questions. Again, going back to what we talked about previously about the evolution of eusociality, right? Because one of the first things that happened during eusocial evolution is that you had the split between reproductive queens and non-reproductive workers. So how reproduction is regulated is a big question. And that's actually something we've been also studying in the clonal radar ants. We published a paper about that a couple of years ago.
Yeah, and in the book, so you kind of use these behaviors to also, I think open up a really important part of the way you think about this, which is whether some of these traits or behaviors are adaptive or not adaptive, or how do you, what would be a way of gaining evidence that something might be adaptive versus, sort of non-adaptive explanations where you just see biological complexity for the sake of biological complexity. And so are there specific, some of these phasic, the rhythms, are these like sort of, the right places to look for adaptive sort of outcomes? Or you mentioned is it the behavioralist, I'm forgetting the name, but the notion of proximate versus ultimate explanations for behaviors.
Tinbergen.
Yeah.
Yeah. It's an interesting question, right? Because because you have, as I said, like, there's some army ants that are phasic and others that are not phasic. Mm-hmm. And it's not intuitive why sometimes, like saying that something is adaptive is easy, but you don't really know why, right? And they're both army ants. Why would you be phasic under some circumstances and non-phasic under other circumstances? It's a hard question to crack, right? Because you can't really experiment with army ants. What you maybe would want to do is something like an experiment where you, in the field, disrupt the phasic cycle of a phasic species, or maybe force a non-phasic species to emigrate regularly or something like that. And that you can't really do. So we did once, we approached this with like a modeling approach. And the idea here was that, maybe if you're like an army ant, it's basically if you're like a regular ant, right? You send out a few workers and they can probably find some food, right? And the more workers you send out, the further they have to travel, and at some point it doesn't become it becomes less rewarding. But that kind of the it plateaus off. So you can kind of haul in kind of a stable amount of food every day, basically. And so if that's your life history, it might be good to also kind of always have like the same numbers of larvae in the colony, roughly, right? Because you always bring in the same amount of food. So kind of your development is or your supply is pretty stable. And so you kind of want to spread larvae out as much as you can. In army ants, it's a little bit different, right? Because if you just send out a small raiding party, they're not going to be successful, right? They have to overwhelm other ant colonies, right? They have to break into like a well-defended social insect colony. But if they manage to do that, then the bounty is plentiful, right? They can plunder the whole nest, and they bring back lots of prey. And so here it's more like an all or nothing scenario. Like either you're all in or you don't have to bother at all, right? And so if you're that kind of organism, it might be better to concentrate larvae kind of all at once. So you raise a lot of larvae and you raid and you bring back a lot of food. And then when the larvae pupate, you can take a break, basically. You can stop the costly raiding and you just let the larvae pupate and develop. And so you kind of go back and forth between the two. And why that might work for some army ants but not others is a little bit an open question, I think. One idea here is that it depends on your prey spectrum, right? Like many army ants and especially the phasic army ants, they seem to be social insect predators. So they are really under this scenario that I just described, that they have to overwhelm well-defended ant and termite colonies and so on. The army ants that are not phasic, and some of these African driver ants are actually not phasic, they usually have much broader diets. So some of them even feed on plant material and pretty much any invertebrate that they'll find. They feed on mammal carcasses and they're pretty unspecific in what they take. And if that's your diet, you might actually, even with you might have like a more like constant food supply, right? And even with like smaller foraging efforts, you might still be able to bring back food. And maybe under that scenario, it kind of tips the balance towards not being phasic. But that's purely speculative, right? It's nothing that's easily testable experimentally, unfortunately.
Yeah, no, absolutely. And how does being nomadic tie into this? Or is that also, I mean, these are really kind of complex behaviors and so really difficult to distill, but is that?
Yeah, I think the nomadic behavior is independent from the phasic behavior, right? Like all army ants are nomadic, whether they're phasic or not. I think the nomadism really has to do with this predatory lifestyle. So, if you clean out your environment, you don't have to go out and ransack a village if you've just sacked it the day before, right? And so you have to move on, basically. And nomadism also seems to be common in these species that are related to army ants, but are not classified as army ants. So that seems something that evolves pretty early on along the trajectory to becoming an army ant. And I think it's mostly related to the predatory behavior.
Gotcha. Yeah, so it's really interesting, these bivouac-like structures that, as you're describing, being really organized. And so, and kind of on the move all the time, but in a pretty stable situation. So you don't all of a sudden go from 1 to 2. It's a really rare event that you describe of actually making a new colony. Maybe take us through that, some of the colony dynamics.
Or when colonies reproduce, you mean?
Yep.
Yeah. So again, like army ants are very, very unusual compared to other ants, right? Like maybe you notice, like sometimes in the spring you go out and there are all these winged ants suddenly like crawling around on the pavement or something. That's how normal ants do it, right? They produce reproductive, so winged queens and winged males, and they send them out on a mating flight. They mate, the males die afterwards, the queens drop to the floor, they shed their wings, and then they start digging a little hole in the soil, and then all by themselves they start laying eggs, and then they slowly build up a colony again, right? And when the colony is big enough, they start to produce new young males and young queens. So if you're a predator, like a mass predator, like an army ant, that doesn't work, right? Like you can't have a single queen laying a few eggs and then you have a colony of 1 queen and 3-4 workers. They can't go out and retrieve food, right? That's impossible. And so you can't afford to go through this colony founding stage, basically. And so what army ant colonies do instead is they grow to very, very large sizes, and then they simply split in 2. And so for like a brief period, they switch from raising workers and growing more to raising young queens and young males. And But what they do is they raise a few thousand males usually, but only very, very few young queens, usually only about 5 or so. And then the males, when the colony splits, one of the 2 colonies often retains the old queen, and then the other colony gets one of the young queens, and the other young queens are left behind to die usually. And then they kind of split, they emigrate, they move apart, and then the males walk in the emigrations for a few nights, and then they take off on the wing and disperse. That's how it works in S. dombucellii, for example, in S. hamedum. So again, it's very unusual biology for an ant. And as a consequence of that, also the queens are permanently wingless. They are massive, partly because the colonies are so big, right? They're among the biggest ants in the world. Like the queens of African army ant, driver ants, they measure up to like 6 centimeters or something in length. So they're massive. I actually have one here. I can show you.
Oh wow, yeah,
in a jar.
Also, some great photos in the book. So I'm just scrolling through of the queens, and I mean, my God, the morphology difference between.
So let's see whether I can do this. So here in the front, you see worker ants of an African driver ant, and then here, this is a queen.
Yeah,
you see this?
Yeah, yeah.
Doesn't look like an ant, right? Here, this is an army ant male from the same species. These are called in Africa. They're known as sausage flies because they're so chunky and big. Those you can collect at lights at night. The queens you hardly ever see because they never leave the colony, right? So you really have to, either you see her on an emigration or you have to excavate a whole nest.
So the colonies where you mentioned one queen, lays a few eggs, and how do they feed themselves? Because you said the army ants can't do that because there's too many of them. But everyone has to. Well, I guess you said they just split in half. But I'm just curious. These other ants that start really small, how do they take care of themselves?
So the young queens usually come with a lot of reserves that they can use early on. And some species, the young queens actually already go out and forage even before they have workers, and then they only stop foraging once the first workers have it closed, once the first workers are born. But then, depending on what they feed on, if you have like 3 or 4 workers, those can go out and bring back, like scavenge or bring back seeds or whatever that particular species feeds on, right? But of course, you can't with 3 workers attack like a different ant colony.
So the one outcome is you split the colony or build into 2, but the other outcome obviously is that it doesn't sustain and the colony is vulnerable. And I guess because you have multiple queens but not emergency queens, that some of these army ants might be vulnerable to these colonies dying. How does this unfold?
So you don't have multiple queens. Like an army ant colony only has 1 queen. Mm-hmm. It's only doing this process of colony fission where they raise kind of a handful of queens. They can't raise emergency queens, so that's it's very different actually from things like honeybees, for example, where if the queen dies, they can usually replace her by raising an emergency queen. Army ants can't do that. So if they lose the queen, the colony is pretty much doomed. And interestingly, what they often do is that they merge with other colonies. So they have this cool behavior when you remove the queen experimentally, or when she gets lost during an emigration, for example, the colony develops what's called backtracking behavior. So instead of the next day after the emigration, instead of like developing an emigration column in the other direction to get further away, they backtrack on the previous night's emigration column. Kind of, it looks like they're really looking for their queen. So they kind of, somehow they know that the queen is missing. But if they can't find the queen, usually after a few days, they kind of make contact with another colony of the same species, and then they diffuse. And that's, again, it's something that's a little bit hard to interpret, right? Because it almost seems like an altruistic act, which you wouldn't expect between 2 colonies that are not related.
Yeah.
But it could be it could be kind of a remnant of. Maybe it is adaptive in the sense that right after colony fission, right, say the young queen is somehow not fertile and she can't reproduce, but the workers realize that too late, then there's still this kind of umbilical cord to the old mother colony, and they can go back and fuse again with the colony that they came from.
Yeah, interesting.
So maybe that's where this behavior comes from, but it's a little bit, again, hard to know. Right?
Sure. Yep. So, and for all these behaviors, we're talking about kind of colony, all these things are unfolding on a colony level, not an individual level. And so you bring up this framework called inclusive fitness theory as a way maybe of trying to tackle these kind of community-based or colony-based behaviors. Maybe help us out a little bit with that, how that applies to ant societies. I'm probably using that word wrong. Ant colonies, not societies probably has is that okay? Okay.
Well, so inclusive fitness theory is this idea that your evolutionary fitness is not only determined by your immediate offspring that you leave behind, right? But it's also determined by the offspring of your relatives, because you your genes essentially can gain fitness through related individuals that have some above average random probability of carrying those same genes by common descent. Right? And that's a very powerful idea in evolutionary biology that actually goes back to Darwin, but then was formalized by Bill Hamilton in the 1960s. And it's very, if you work on social insects, use social insects, right? That's something you have to kind of internalize, I think, to understand how these societies evolve, because again, one defining feature of an insect society is this reproductive division of labor, right? Like some individuals are just sterile, they just don't reproduce. And clearly that's a trait that can't be adaptive at the individual level, right? That's the ultimate non-adaptive trait, right? Like sterility, that's an evolutionary dead end, right? Yeah. So this can really only evolve via inclusive fitness, by indirect benefits to these altruistic non-reproductive workers via the queen germline, basically, right? So they're closely related to the queen, and so they help the queen reproduce, and they gain fitness benefits via the offspring of the queen, right? Via the queens and the young queens and the young males that colony produces. So that's kind of inclusive fitness theory 101. But that's also it's kind of the main reason, I think, why ant societies and human societies are so fundamentally different. Right? So when I talk to journalists, they often ask me about, what can we learn from ant societies about human societies? And my answer is usually nothing, right? And you really shouldn't, because that's important to keep in mind, right? Like in ant societies, because you have this reproductive division of labor, all the individuality goes out of the window, right? Now evolution can shape the workers in whatever way it wants, right? They're not these like jacks of all trades anymore. They can completely specialize, and they use a lot of individual functions, right? That a complete organism should have, like being able to reproduce, right? And so it really becomes more like an organism, right? And that's this analogy of the ant colony to an organism or superorganism, right? Where the queen is kind of the germline, and the workers are the somatic cells. And then even within the workers, right, you can have different types of workers, like the soldiers and the small nurse workers and so on. And those are a little bit like different cell types or different organ types, right? So all this individuality is gone in ant societies, right? And they're just integrated in a way that's fundamentally different from a human society. And I think that's important to keep in mind when making comparisons.
I guess the human societies that have tried to emulate that have all failed, right?
Yes, you'd be like a very authoritarian society, right? And that, of course, doesn't work if you have individualistic tendencies among the individuals, which is obviously a good thing, right? And
yeah, some failed experiments there. Hopefully, I won't try to attach that too much to our recent history, but really fun, I think, to consider then in these scenarios like this, where you start to see this inclusive fitness. Then what are the consequences? How is that encoded in a genome? Or what are the consequences to genome evolution? That sort of goes back to that workshop where we met up a few years ago and thinking, and again, sort of kind of a frontier of moving forward some of these ideas and thinking about what are the systems the clonal raider ants or other social insects, honeybees, et cetera, where you can start to get a footing and start looking at this. And yeah, I think it's really exciting.
It's an interesting question. Like I personally, I don't know now, maybe you know more about this than I do because you've been actually involved in this discussion back then. my impression is, well, first you have to keep in mind that in the genome, you still have to encode all the functions, right? Because the genome of the queen and the worker are the same, right? So you have to, just because the workers don't have wings anymore, you still have to be able to make wings because you need to be able to make a queen, right? So that sets some limitations, right? You can't just split the genome up into the different functions. And so if when people started to sequence the first ant genomes, I think they were very excited about this idea of you look at the genome and you understand what makes them eusocial, right? And it's a little bit like when you compare the human genome to the chimp genome, right? Like people got very excited about it, but you look at it too and they kind of look the same, right? And the same is true for ants. I think there's one thing that really stands out, and then that is this massive expansion in odorant receptor genes compared to solitary insects. That kind of goes back to this, what we discussed earlier on with Orco, right? And that's a signature, I think, of becoming ant eusocial, right? It's a peculiar thing, but because they evolved this very complex pheromone language, and I think they did that partly, at least on the receiving end, via this expansion in odorant receptors. That's really the one signature that jumps at you in ant genomes. Other than that, it's more subtle, right?
Oh yeah, certainly a story of gene regulation, right? So how, and as manifested by those totally different morphologies between a queen, a worker, a soldier.
Yeah. So those are epigenetic effects, right? And that's clearly very interesting to figure out how that works. We had this one paper a couple of years ago with the clonal raider ant, where we kind of look at how this cycle is regulated on a molecular level. And what's really fascinating is that it seems that like insulin signaling plays a key role here. So insulin levels are high in the reproductive phase or the stationary phase when the ants lay eggs, and then it's low in the brood care phase or the nomadic phase. And what's cool is that the insulin levels in the adults actually are regulated by the larvae. So you can do these experiments where you have adults and you completely control the nutritional state and everything, but then you either add larvae or you remove larvae, you switch larvae between the two treatment groups, and immediately you see like insulin levels go either up or down. And of course that then has all kinds of downstream regulatory effects. So it's kind of just because you talked about gene expression, right? It's kind of this cool example where this very ancient metabolic pathway that usually senses nutritional state of the organism has been put partly under social control, right? To now kind of regulate almost like the metabolism of the superorganism, right?
Really exciting.
That's kind of one I find kind of a nice example of how things get rewired, right? But it's a very specific example.
Yeah,
and you probably wouldn't see it just from looking at the genome sequence.
Oh, absolutely. Yep. So then you kind of close on a really fun note, which is the circus around the swarm. So these are all the social parasites that are, when a big army ant colony gets together and has a raiding swarm, they're not alone. All kinds of other species are getting in on the action in various shapes and forms. So maybe tell us a little bit about that. Who are these social parasites and what are they doing?
So that's another thing that army ant colonies are famous for. And again, it's related to their massive sizes, right? So if you have a successful lifestyle and you have like a lot of resources, of course you become attractive to parasites, as we all know, right? And in army ant societies, they have been infiltrated again and again by these social parasites, and those range from anything from like little fort flies to actually snakes that live inside the army ant colonies. And so they're more diverse. You have social parasites also in other ant species and also in termite species, but in army ants, they're just particularly diverse. And that's related to these massive colony sizes and all the different kind of ecological niches that an army ant colony has to offer, right? So it's like this little ecosystem in itself. And so you get this microcosm of these social parasites. And what's really cool is that, these social parasites, they have to move with the colony. So they have to keep up with the colony. So you can sit there at the colony immigration at night in the rainforest in Costa Rica with your headlamp and you just watch the ants walk past. And every now and then you see this weird thing, like a beetle or a millipede, some strange animal that just like walks along with the ants, or some of them ride on the ants or being carried by the ants. And that's where this traveling circus comes from.
And the ants don't bother them.
So it depends on the species. there's a lot of strategies that these social parasites have to infiltrate the army ant colonies. Some of them are very specialized. Some of them mimic the ants, so-called Wasmannian mimics. They really look like ants, even though they're beetles, for example, or wasps. They acquire some of these chemicals that the ants use to tell self from non-self. They have like these particular hydrocarbons that they use as colony signature odors. Some of these social parasites are very good at acquiring those, and then they really can fly under the radar, and the ants don't bother them. But then there are others that mostly those that feed on the ant brood, so they're kind of detrimental to the ant colony. And those often have evolved these very protective morphology. So they're either like tank-like or shaped like a drop so that the ants can't pick them up. They're like very slippery, and they can retract the legs and the antennae. And so they don't really have anything for the ants to bite. And those you see as well. And those are kind of, they always look like a little bit on edge. Like as soon as the ants come, they kind of tighten up. But then the ants can't do anything. And as soon as the ants walk on, then the beetle kind of extends the legs again and keeps on walking.
But these parasites take something from the ants. That's why we call them parasites, right?
Yes. So in the best case, they feed on the ant prey. So they're more like kind of commensals, and they probably don't have like a massive impact on the ant society, a massive negative impact. But then others feed on the ants themselves or the ant brood. So, for example, these snakes that I mentioned, they actually prey on ant brood, and the snakes are big for an ant, right? They're like, I don't know, 30 centimeters or something. Mm-hmm. And if you have a snake like this in your colony, it just gobbles up your larvae, right? Or your pupae.
One of the kind of intrusive examples that caught my eye is this clown beetle. You've got a great picture of this. It's just a army ant, and it looks like it has two abdomens. So what's going on here with this?
Yeah, so this is one of the species we discovered doing our studies at La Selva Biological Station in Costa Rica. And that's a beetle that's specialized on one particular army ant species, Eceton mexicanum. And the way that beetle follows the emigrations is that it rides on the ant. So it kind of clamps with its mandibles on the ant waist, and then retracts the legs, and it kind of looks like this sphere, a little bit like the ant abdomen. So when you see the ant in profile, it looks like it has two butts, basically. Yeah. And we found that beetle hadn't been discovered, even though it's quite remarkable, right? And the way we found it is, Christoph and I were sitting at an army ant emigration at night of that species, looking for these guests, these myrmecophiles. And the ones that walk freely, they're more easy to spot. But the ones that are actually on the ants or under the ants, they're really hard to pick out. And this one, it was just that every now and then there was one ant that walked past that reflected the light from our headlamp slightly differently. And so we started wondering what that was, and then we started to collect some with our aspirator. And as soon as you have them in a falcon tube and you shake it a little bit, the beetle kind of falls off and starts walking around, and you see that there's actually something else in there. And then once you know what you're looking for, once you have some search image, it actually turned out that there are quite a few of those in each colony of that species that we looked at. So they're not rare; they're just hard to find. But once you know they're there, you'll see them.
What a cool discovery! And then since you discovered it, I guess you got to name it. So help me out here. This is Nymphister Kronauerii.
So I didn't name it. Christoph named it with our taxonomist collaborator, and so it was named after me. That's my first official species. And,
that's
and
awesome.
A nice one on top of that. So
I'm
very happy with that.
That's a great achievement.
It's kind of it's a nice story. I think it's one of these things that it's probably hard to sell to a funding agency, but it's one of these discoveries that just makes people happy somehow. We've probably got more press coverage about that than, for example, our orca knockouts, even though scientifically it's a more significant achievement maybe. But the beetle, my sister on the train in Hamburg saw the beetle featured on one of the screens on the train, and these kind of things. Like
you're famous. Yeah, exactly. Yep. So how about at the microbial level? Are there folks looking at parasites that are microbes? I'm thinking about infectious microbes, I guess, to some degree, but also other associations. And what's known there?
Infectious microbes in army ants, people haven't looked into much. But what's quite interesting is that they have quite a specialized gut microbiome, army ants. So some group of ants have specialized gut microbes. Sometimes these ants that feed a lot on things like extrafoil nectars, and like very sugary excretions. They often have specialized microbes that help with things like nitrogen upgrading. The army ants have very different ones, and we don't really know much about what they do functionally, but it could again be related to this very specialized diet, right? So they have like a very meat-heavy diet, and maybe those microbes again help in kind of digestion or nutrient upgrading in some ways. But they're very poorly characterized on a kind of on a functional level. We haven't been able to culture them yet, and that would be kind of a first step, I think, to kind of figure out what they're doing and maybe do some metabolomics and
yeah, exactly. Maybe some chitinases hanging out there to digest these exoskeletons. Who knows what could be?
Yeah, so they don't eat the exoskeleton. They usually suck kind of the bodily fluids out. But yeah, it's an interesting topic. I think that's something I've been mostly been involved in as a collaborator. There's Jake Russell at Drexel University has been working on that quite a lot, and we've been sending him samples of army ants.
Sounds like something for you, Nels. Look at some of the viruses that might be there.
Yeah, that's
Send
right.
Nels some ants.
The viruses we don't know anything.
Send Nels some ants. He'll do that.
Just need to get some GFP reporter strains, and we'll be off and running, and then we can go. No, so really exciting. I think we won't keep you forever here, Daniel. We could talk for hours, or do, as we've said, hinted at, do entire podcasts on this. But hopefully, kind of an appetizer. We've just barely scratched the surface, sort of the tip of the bivouac, so to speak, of all of the ant complexity here. Again, amazing camera work from all of your field journeys and sort of Renaissance science here, I would say, going all the way from the field to the laboratory with some of the clonal raider ants and some of those stories, and then weaving that together with a scientific framework. And I think, both as just a science curious person, but also to our audience of grad students and others coming up in science, like take a close look at some of the questions, the biological complexity here, and the tools that are just coming online. I think some incredible opportunities for all kinds of cool careers in science coming around this biology.
So I have to ask you, what kind of camera do you use?
I use Canon bodies. So this is actually all these photos are taken with a pretty old EOS 7D camera that I've had for over 10 years now. I'm overdue for a camera body upgrade is what I'm trying to say.
Oh, there's some good ones out there.
Yeah.
And hopefully you can...
Now all the mirrorless cameras are out for
well, you can...
macro photography, and
you can use your lenses. You can get adapters to use probably have a collection of lenses that you'd want to keep,
Yeah.
It's very cool. I'm glad to hear you. I'm glad you didn't say I use my iPhone. That's good.
No. The trick is always the lighting, right? And I've actually built these kind of apparatuses to like a light box that you can put around the scene. That's much easier than having like on-camera flash diffusers, because those always get in the way and the ants are just not very happy. And then before you know it, you have like a few hundred irate army ants on your camera. So it's a little bit more flexible.
Right.
Way.
So now should we let Daniel go and we can finish up?
Yeah, that sounds good. Although, Daniel, do you have a science pick of the week, or?
Yes, I have thought about that a little bit. So one, I have two science picks. I keep it very short. So one is a very nice book. So Harvard University Press that has published my army ants book. They have like a history of publishing really great ant books. And one that came out this year too is this one here, Desert Navigator by Rudiger Bainer. And Rudiger, he has been studying these desert ants for almost 50 years. And that's kind of his, like the summary of his life's work, I would say. And those ants are really fascinating because they don't use pheromone trails for navigation. They're on the sand mostly, like any pheromones would just evaporate. And they do path navigation, path integration, and use all kinds of other landmarks and odorant cues to find back home. Wow. And it's a very cool behavioral paradigm, and lots of interesting physiology and kind of inspiration for even neuroscience studies in the future are in there.
Yeah, it's cool. I mean, one thing. So is it something estimated to be something like in the order of 14,000 species of ants? Did I get that right?
Yes. Yeah. That's only the described ones. They're probably more like closer to 20,000 probably.
Yeah.
Total, I'd say.
No, and one of the things that grabbed me is just in some of the phylogenetic trees that you show, the illustrations that you have, is, where you're pinpointing in on and really focusing on a couple species, but these are like at sort of family level, subfamily level, in the Doralineae, but then you just keep going out the tree and there's just, story after story after story, including like what you're saying here with the desert ants. It's just incredible, the diversity here and the biology even among the ants just kind of blows the mind.
Yeah. No, it's a good field to study. It doesn't get boring. A little bit like viruses, maybe.
Oh, for sure.
Yeah, there's a lot of diversity there too.
Sure.
Although, Vincent, there's no virus named after you yet.
That's okay. You don't need it too hard.
Oh, okay.
Did you have a second pick? Did you have a second pick, you said?
Oh, yeah. There's another great book that I really like that it's a little bit old. It came out in 2017. It's basically, it's a new edition of a very old book by Maria Sibylla Merian called Metamorphosis Insectorum Surinamensium. It's the metamorphosis of the insects of Suriname. And so she published this book in 1705. So really old. The new edition was released because Merian died 300 years ago in 2017. And so I picked her because she provided kind of the first illustration of an army ant raid, of army ants and leaf-cutting ants on a guava tree. And she was kind of really one of the foremost female scientists of the 17th century. And she, back in those days, like people used to kind of depict animals mostly like in isolation, kind of as kind of collection items. And she really drew animals in kind of a natural context, like with plants and other species they would interact with. So she kind of, I think, anticipated this famous metaphor of Charles Darwin of like nature as a tangled bank, kind of by probably a good 150 years or so. And the book is just beautiful. It's like in the original size. It's a very big book, but her watercolors are just amazing. That's really something that's worth checking out.
Yeah, we'll add the links to both of those books. And that was one thing that we probably skimped on a little bit in our discussion today was some of that history, the scientific history of stepping through a lot of the, which is also a thread woven in to Daniel's new book as well. So my science pick of the week is NPR Science Commerce. This is a science communication collective. This just came on my radar maybe a few weeks ago. It's run by Joe Pelka, who you'll hear if you tune into, All Things Considered or whatever on NPR in the afternoon, you'll hear him in his stories. Maddie Sophia and Burley McCoy. They describe this as a community of scientists and engineers that includes undergrads, grad students, postdocs, and faculty interested in improving their science communication skills. And so there's a website, we'll put in the link, and currently something like 1,600 plus participants or science commers who are out there kind of approaching this. There's a writer's program, chats with mentors, other resources to build a network around science communication. And so, sometimes people ask me like, oh, how did I get into this science podcasting? And my answer generally isn't very satisfying. It's sort of this weird collision of hanging out at a meeting and running into Vincent. And so, it's probably not the like totally normal way of getting into science communication. Not to say that there is a normal way to get into science communication. And so I think this is a great resource where if you go there, you can apply I think it's just an email and a connection to join the Science Commerce. So that's my pick of the week here.
I guess not everyone can run into me at meetings right now.
Prolific science podcaster. Yeah, that was a good chance encounter, a good collision.
Cool. Very cool.
How about you, Vincent? What's your pick this time?
A very, very quirky website, which is called The Vintage Beauty of Soviet Control Rooms. And it's just a series of photographs, quite old, of people working in these massive control rooms with all kinds of dials, and they're all analog, right? Pre-digital, and switches. And they just have this incredible beauty to them that we don't see anymore, right? Dials, stuff. It's great. Just check it out. The Vintage Beauty of Soviet Control Rooms. It's a little bit science, because it's.
I'm scrolling through.
They're really controlling reactors or power plants or stuff like that, big, massive things. And some of the people in them, they have white coats on and things on their head and so forth. It's funny. We don't have this anymore. It's all modern, but this is kind of a cool thing.
Yeah, very interesting. Sort of the collision of technology and art almost somehow. Yeah.
All right, that's TWiEVO 62, microbe.tv/TWiEVO. Questions and comments, TWiEVO at microbe.tv. If you like what we do, go over to microbe.tv/contribute. And you can support us kind of like the NPR model, right? We don't do ads, but we survive on your support. Our guest today, Daniel Kronauer from Rockefeller University. Thanks so much for joining us. It was great.
Thank you for having me. That was great.
Good luck with the book. It was just published, right?
Yeah, in October.
Well, now you get wealthy and you can retire, right?
Let's see how that goes.
Nels LD is at cellvolution.org on Twitter. Early Bird. Thanks, Nels.
Yeah, thank you, Vincent. Good to be together. And thanks a lot, Daniel. Really fun to hear about sort of behind the scenes and with this great book. Big recommend. If you're looking for something on your holiday shopping list, your gift recipient will not be disappointed.
Thanks, Nels.
I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious.